Hello everyone, we are Chromosome Biology Lab in Kumamoto University IMED. Recently, we found a novel gene important for genetic diversity and also for sperm production. Meiosis is a specialized cell division that generates sperm or egg. During meiosis, genetic information is exchanged between maternal and paternal chromosomes, which gives genetic differences to the next generation. Meiotic recombination is initiated by DNA breaks as normal process. However, DNA break is nothing less than DNA damage that is a threat to the cells. Thus, DNA breaks must be repaired immediately. In this study, we discovered a novel gene that plays a crucial role in such repairing of DNA damage during meiotic recombination. Previously, we discovered meiosin that act as a switch from mitosis to meiosis. Meiosin turns on hundreds of the meiotic genes to start meiosis. C19orf57 was one of those genes controlled by meiosin, but its function was unknown. In this study, we analyzed the function of C19orf57 during meiotic recombination. PCR tests indicate that C19orf57 gene is expressed in testis and ovary, but not in other tissues. To our surprise, by mass spectral analysis, we found that C19orf57 binds to the breast cancer suppressor BRCA2. These data suggest C19orf57 and BRCA2 function together in germ cells. Here's another evidence showing the cooperation between C19orf57 and BRCA2. This microscopic image shows chromosomes in spermatocytes that is undergoing meiotic recombination. Blue lines indicate chromosomes. Green foci indicates C19orf57. Red indicates BRCA2. We see C19orf57 comes to the DNA damage sites. BRCA2 also comes to the same places. This image tells us that C19orf57 comes to the DNA damage sites and recruit BRCA2 to the DNA damage sites. Here's the model that explains our finding. DNA breaks are introduced on chromosomes. This triggers meiotic recombination. But such DNA breaks are nothing less than DNA damage for the cells, so this must be repaired immediately. The molecule C19orf57 that we newly identified, together with the breast cancer suppressor BRCA2, works in this DNA damage repair during meiotic recombination. We disrupted CR19orf57 in mice by genome editing technology. Without C19orf57, testes become smaller and male mice cannot produce sperm. This makes male mice infertile. In summary, we discovered a novel gene, C19orf57, that is required for meiotic recombination. C19orf57 recruits breast cancer suppressor BRCA2 to DNA breaks, and the impairment of C19orf7 led to male infertility. Our finding sheds light of the mechanism of infertility. We would like to thank our collaborators and the funding.